Welcome to the first key area in your higher biology course, and this is going to be looking at the structure and the organisation of DNA. Now this structure that's here should be quite familiar to you. You learnt last year that DNA was a double spiral helix, that it had a backbone, and you may have learnt that it was a sugar phosphate backbone, and that it was comprised of bases that are forming this latter part, the bases being adenine, thymine, cytosine and guanine. Now looking at this structure in more detail, I've got a note here. It says that the DNA molecule is comprised of two chains of nucleotides. Now nucleotides are made up of sugar and phosphate and a base, and that's what forms your sugar and your phosphate is what forms your sugar phosphate backbone, and attached to that is your base. So your DNA is made up of these nucleotides all joined together, and we're going to look at these nucleotides in more detail. Well, here is the basic structure of a nucleotide here. It's made up of a sugar, and that's a deoxyribose sugar, and a phosphate group, and then a base. And that base can be either adenine, thymine, guanine or cytosine. Now you'll notice that I've got here that I've got something here called a 5' end and a 3' end. So it's with this little comma that is here, it's pronounced prime. So we have this 3' at the deoxyribose sugar and a 5' at the phosphate end. Now we're going to look at the structure more closely to just work out what these 3' and 5' are representing. Now you don't need to know the nucleotide structure in as much detail as this, but it helps you to give you an idea of where this 3' and 5' is coming from. And they are representing where the carbon atoms are on the sugar. So if I look at the sugar here, here's a carbon here, number 1, here 2, carbon 3, carbon 4 and carbon 5. So that's where we get the 3' from, and the 5' is representing where these carbons are here. When we take one of these nucleotides and then we join them together to a row of other nucleotides, then what happens is we form a strand of DNA. So here you've got one nucleotide here, and then you've got another one joined to it here, and this will carry on so forth to make a long chain of nucleotides called a polynucleotide. This part here is what forms the sugar phosphate backbone, and these are the bases, whatever those bases happen to be. So when we take all these nucleotides all joined together, that'll form one strand, and then you'll form another strand on the opposite side, and you'll make your DNA. We'll look in a wee bit more detail at these strands in a moment, but I want to just quickly remind ourselves about the bases on DNA, which are adenine, thymine, cytosine and guanine. And you'll remember from last year, these bases are described as being complementary to each other, where your adenine base always pairs up with your thymine base, and your guanine always pairs up with your cytosine. The bases are joined together by hydrogen bonds, and so when you end up with your two strands, the sugar phosphate backbone and the bases, remembering that structure of the nucleotide right the way down, and you remember you've got your base pairing, and this base sequence that is here forms a genetic code. In this slide here, I want to look in more detail at the actual structure of the DNA strand. And what you're looking at here is one strand over at this side here, made up of the nucleotides. So here's one nucleotide here, and here is the next one, and so on and so forth. And over on the other side here, we've got the other strand. Now, when you look at these two strands, I'm just going to adjust the size of this slide here a little bit to look at this. When you look at these two strands here, you will see that this strand over at this side here, you start with a 3' end here, and you've got the 5' end here, and the ribose sugars are facing this way. But on the other side, you'll notice that the 5' end is here, and the 3' end is up here. The ribose sugars here are facing the other way. So the double helix strand, these two strands here, are described as having two anti-parallel chains of nucleotides, because one side goes from 5'3", this side here, and the other side goes from 3 to 5. So if you look at the 5 to 3 direction, here it's going this way, but on this other strand it's going this way. So the strands are going in opposite directions from each other. You could think of them like that. And that is described as an antiparallel structure. We can look at that antiparallel structure again in this diagram here, which actually is showing you the actual structures of the nucleotides. As I've said before, you don't need to know these chemical formulas that are here, but it just helps you to see what's happening. So over at this side here, you'll see on this molecule that is here, you've got the 5' up here and the 3' here, the 3' end. So your 5 to 3 is going this direction. On the other strand, over at this side here, you've got your 5' end here and your 3' end up here, and 5'3" is going in that direction. And over here, you can see the whole of their double spiral helix. You've got the two backbones that are here. These are the sugar phosphate backbones. And then from over here, you'll see the bases that are here, and you've got the hydrogen bonds between them. So when these hydrogen bonds are formed, like over here, then you start to get the two chains twisting to form the double spiral helix. It's important to remember these two strands going in opposite directions, because when we come to look at DNA replication, that has implications on how the replication is carried out depending on which strand we're looking at. Now, I don't need to read this slide out to you. You can read it over yourself. These are just the key concepts around this area.